We are ready, ma'am. Darshi, ma'am?
Yeah, okay.
We
I
are ready.
think, yes,
Yeah,
Parth, you can start.
yeah. A very good evening and greetings to one and all. I, Dr. Parth Pandya, on behalf of Organizing Committee NUV, welcome you all for the NUV Biotechnology Conversation Series. It gives me immense warmth and great pleasure to grace all of your presence for today's session. As the world has seen several greatest pandemics in its history, COVID-19 is considered to be the deadliest one. If we see the world statistics, WHO has estimated that 24 million people across the world has been affected by SARS-CoV-2. In this regards, to spread the awareness on COVID-19, School of Science, Navrachna University has organized a panel discussion on the topic COVID-19 polymerase chain reaction remains the final answer. At the beginning, I invite Professor Nilay Yagnik, Provost, Navrachna University, to give the opening remarks. Please, sir.
Thank you, Dr. Parth. Welcome to Navrachna University. And on behalf of everyone here at Navrachna University, I wish to thank Dr. Mayuranki, Mr. Mohal Sarabhai, and Dr. Rebecca for speaking to all of us today and enlightening us on an extremely important and a very relevant topic. Everyone here, the general public, not just the technical people or the pure science people, but the general public is very concerned about the current disease that is there, COVID. And the aspect of testing, therefore, or how accurate is the test, is also a great area of concern for everyone today. And therefore, when you see, multiple ways of testing exist. And this particular aspect of testing, which is PCR, is something which is supposed to be an accurate method. So today, we have a very interesting discussion, a conversation series as a part of the Navrachna University School of Science Biotechnology Conversation Series on the accuracy of PCR and why possibly PCR is the best solution for accurate testing of COVID. Navrachna University has always been at the forefront in teaching and research. We always conduct research, we always teach things which are extremely relevant and also at the next generation. And this particular topic, therefore, is very much in line with our vision of being at the forefront of both teaching and research in an interdisciplinary framework. So I thank you, Dr. Mayuranki, Mr. Mohal Sarabhai, and Dr. Rebecca for coming over today and giving us a very nice session. And we really look forward to your session today. Thank you.
Thank you.
Thank you very much, sir. Moving towards the session, I feel honored to welcome and introduce Dr. Mayuranki Almola, who will be the moderator for today's session. She has done her master's in biological sciences and PhD in molecular biology from Stanford University, USA. She is having a rich research experience for more than 24 years and is actively engrossed in the diagnostic research. She has worked in several reputed diagnostic companies. To name a few, like Silico Sciences, Symbiosis Biowares, Bioimagine, and presently, she is looking after the Strategic Alliance Indian operation for co-diagnostics. In terms of research, she has designed and guided several major projects. For instance, the annotation and curation of receptor tyrosine kinase and several other proteins in oncology pathways. She has developed a database for several specific disease areas with pharmacokinetic ADME information, and the first major biomarker validation project with GeneProbe, and the list goes on. With these few words, I invite and request Dr. Mayuranki to introduce our eminent panelist, Mr. Sarabhai, MD and CEO, Essence Group USA, and Dr. Rebecca, VP Co-Diagnostic USA, and begin today's session. Please, madam.
Thank you, Dr. Parth. It is a pleasure and an honor for both Co-Diagnostics and CoSara to be invited to be a part of this distinguished In Conversation series. I will start the evening by introducing our both very dynamic speakers who are going to participate in the conversation. And Dr. Rebecca Garcia is the Vice President of Product Development at Co-Diagnostics Inc. Dr. Garcia has a very crucial role that she plays due to her extremely impressive background. She was one of those students that finished her PhD in record time. She has a degree in human genetics from Clemson University. She has worked on early designs and the technology that Co-Diagnostics has patented and is responsible for the earlier iterations of many of our tests that include HIV, that include dengue, hepatitis B and C. And also in her earlier years, she has worked on some of the cancer markers too. Dr. Garcia has been there from the early inception as well as the successive years of Co-Diagnostics' life story, and is currently also responsible for launching our COVID-19 test. The other speaker is Mr. Mohal Sarabhai. Mr. Mohal Sarabhai is a mechanical engineer by training. He obtained his degree from the University of Madison, Wisconsin. Subsequently, he has been solely responsible for the resurrection of the Ambalal Sarabhai enterprises. Under his stewardship, there are 3 companies right now, and which are, one is in the effervescent formulations that's called Voivantis, fermentation industry, which is taken care of by Symbiotics, and he also heads the joint venture between Co-Diagnostics and Symbiotics called CoSara Diagnostics Private Limited, which was his foray into molecular diagnostics, stepping out of the pharmaceutical zone. I will start the proceedings with the first set of questions, which will be directed towards the technical part of the COVID design, and they will be directed to Dr. Rebecca Garcia. Subsequently, we will bring the general vision and the atmosphere that we can anticipate in terms of COVID, and they will be directed to Mr. Mohal Sarabhai. So, Rebecca, when this whole pandemic started and the world started reading about it in December, can you share your thoughts on the WHO recommended 3-gene or 2-gene design that was originally prescribed in what they thought would be a more confident identification of the COVID-19 virus?
Yes, so the WHO provided interim guidance. They first released it in January of 2020, somewhere around January the 10th. And the interpretation, I believe, from that guidance was somewhat misinterpreted and mishandled. In such a pandemic, as everybody started going into a fear mode, we have to step back and we have to look at the context in which that was released. From what I could read about the interpretation of those guidance, the WHO never specifically required a COVID detection requiring 2 or 3 genes. They simply stated to start with the broad testing aspect, and in hopes that we could start collecting data from that point and more specifically define, what specific test or gene is going to detect the COVID-19. So from that context, what they were trying to do was stimulate people and scientists to collaborate and promote the sharing of data and genomics as it was being discovered from more testing of the COVID, and especially as it started to spread around the world. Specifically though, from the WHO, it clearly stated in the guidance that in laboratories that have validated broad range coronavirus RT-PCR, nucleic acid testing may be available, but insufficient information is available to guarantee proper detection. And then several times in the guidance documents, they also mentioned that samples must test negative repeatedly from 2 sequential samples to confirm the clearance of the virus. And then they also mentioned throughout the guidance documents that in the future, alternative nucleic acid tests would be developed with a better speed and accuracy, or even simplicity, that people should, move towards using instead of a broad test. But unfortunately, I think with the media hype and all of the fear that was created from media, the full intention of molecular testing was completely missed. And then people started going to the governments and promoting a 2-3 gene model. And the governments then adopted the 2-3 gene model, which was a broad range testing instead of using a rapid simplistic method of a confirmatory gene like the RDRP gene.
So then the functional genes that these tests seem to be flashing everywhere, either E or N, S or M. So is there any cross reactivity in the corona family due to these? And would that impede a test design?
Yes, so let me share my screen and I will show the homology between the genes. Okay, so these genes, the E, M, N, and S, they all code structural proteins. And from the screen here, I'm showing, this is a figure 3 from 2 scientists that had published in 2020 the structural arrangement of the COVID-19 versus the SARS virus and the MERS virus. And this was the best depiction that I could find in the literature to actually show you how these genes of S, E, and M, they're very similar across the different strains of beta coronaviruses. As you know, the COVID-19, the SARS, and the MERS, they're all in a family of beta coronaviruses, and there's 6 total that infect humans. So there's a lot of homology that's the same across these. Now, structurally, the genes of course, code the proteins, and the proteins have only, you can see here, 3 amino acids different between the MERS and the COVID-19. The genomic material is pretty much the same. It can be anywhere between 50 to 99% homology between the different genotypes. But the protein structure themselves, the proteins are in different arrangements per the type of virus. So that's where you can have a lot of cross reaction in the tests that are developed, especially if the tests are developed for a gene that is a structural gene versus something that is specific to the COVID-19. And what the literature and the data that's been collected to date shows is that the RDRP gene is highly conserved in the COVID-19 and is specific to COVID-19. It is not necessarily shared by the other beta coronaviruses. There has been one publication that I could find in the literature that where they tested all the different types of genes in like a big multiplex assay. And what they found was that the N gene and the RDRP gene actually performed better than detection using the S, the E, and the M genes. But overall, you know, the beta coronaviruses, the sequence homology with COVID and SARS is somewhere around 77%. So they got 77% of the genome that's shared between them. And then MERS is a little bit less, even though its protein structure is 3 amino acids different for the spike protein. The MERS and the COVID-19 have about a 50% homology in their genome sequences. So the amount of cross-reactivity really depends on the target area in which the test is designed. So where the primers and probes are targeting in the genome, which gene they're targeting, and actually the amount of variation that's within that target area. So it's possible that a test developed early in the COVID outbreak could now show an increase in false results over the time due to the buildup of variation in the genome under the primers or in the target area for the design.
So is this what went wrong with the, and is this what compromised the CDC primers that came out and they had to withdraw the earlier design?
So I believe so. You know, using the primer set that the CDC published originally, an in silico analysis shows over 262 mutations within the target area. So let me show you an example of that. I'm going to share my screen again with an example of where we can blast the primers or probe to the national database. Today it has over 22,000, excuse me, 22,000 COVID sequences in the database. And so if we take that and the primers or the target area and we blast it into that database using an InBlast server, you can see here all of the different COVID sequences that have been published to date, and all these little dots along the way are mutations that have built up either through the virus naturally mutating over time or through the spread of the coronavirus, you know, as it goes to different populations, it collects mutations. And of course, environmental and genomic mutations can play a role in this. But you can see in this area, the green and the red areas over to the right hand of the screen have a larger buildup of mutations than those towards the left hand of the screen. And so if you do this with the CDC primers that they had published on their website, that's where you can actually highlight that specific area and you can see those 262 mutations. And then what also is important is not only the number of mutations in the area, but also, you know, are there mutations that occur together? So if you have an area that only has like a single mutation point, then that most likely isn't going to affect a primer binding to its target. But if you have more than 2 or 3 mutations that have built up in the same area, that can definitely lead to mispriming events or cross reactions with other types of viruses.
So when there is a constant concern about some of the more rampant comorbidities, as in hypertension, diabetes, can you elaborate on the angiotensin converting enzyme inhibitors or their receptor blockers, which play a key role in the viral entry?
Yeah, so I was speaking with a pharmacist just yesterday about this. These inhibitors or blockers increase angiotensin converting enzyme 2. That's the ACE2 receptor, and it's usually found in the cardiopulmonary circulation. what we've known, and it's even published in the peer-reviewed literature, is that ACE2 is a known binding site for COVID-19 virus. And therefore, in theory, patients taking these ACE inhibitors or angiotensin receptor blockers might be at an increased risk of severe COVID-19 infections. Of course, this can't be confirmed until substantive testing is completed, and we have been testing over this period of time those instances. And I found in the Lancet a study that was published in May, around May the 14th, that collected data from 1,139 COVID cases and 11,300 controls. And it showed that there was a significant higher portion of COVID cases with pre-existing cardiovascular disease than the control group, but no increased risk was observed with the actual inhibitors or blockers. So the question remains, is it actually the medicine that's causing the increased risk that the COVID infection, or is it the pre-existing conditions that result in them taking the medicine that then result in them, having the higher chance of getting COVID or having a higher mortality rate if they do get COVID?
You have gone through the travails of designing a new test, particularly in these high pressure times. And so what, according to you, are the major pitfalls in trying to design a very sensitive test?
So I think
it is a sensitive question, so tread carefully.
Everybody wants the best test that they can get. Everybody wants the high sensitivity and the high specificity. And with some of the new technology that's out in the market, not only technology from Co-Diagnostics, but other groups, they have excellent technology that can accomplish these improvements. But the natural human response is to question everything and resist change. So what happens is we have an excellent test that has a high sensitivity, and in return, people end up comparing it to older methods or older technology. And then they're like, okay, why does your test have a high false positive rate? Well, okay, what are you comparing the test to? If the test you're comparing it to doesn't have all of the improvements that's been made, then how can you say that it's a false positive when I may be actually detecting positives and your test that you're comparing it to is an older technology? It may be built with a different gene. It may have, built in a different area of the genome, which has a higher mutation level. It's not actually comparing apples to apples. You're comparing something completely different, either method-wise or, content-wise, to the new methodology. So I think there needs to be, a better understanding and appreciation for improvements that's been made over time for technology and the ability to have higher sensitivity levels.
So, given that there is so much trepidation when they are facing very high level of sensitive outcome, where you end up dealing with more positives than you would ideally like to, can you reflect a little bit on what, according to you, would be a better quality of probe and primer design? I'm going through these questions right now to bring you back to the more simplistic question later, as in what we are going to do with the antigen antibody tests. So, can you throw some light on what exactly is a quality probe and a primer in detection?
Yeah, sure. So let's start with just the difference between traditional PCR and RT-PCR, because this sort of gives an idea of, the historical models versus a newer technology and how we really can't compare the two. So let me share my screen. Right. And this is just some pictures that I took from a recent article from Park Wan, Choi, and Lee in 2020, where they were showing optimization of primer sets. And yes, this came from their article, but this is pretty much the same across the way. So you can see here on the left-hand side of the screen, if I built a COVID test for traditional PCR, so this is, run out on the gel, so you've got extra costs for running out gels and such. You're not seeing it in real time, so it's sort of, you run it and the banding that you see represents a positive reaction. So the bands you can see here, whenever you have different types of test design and, there's different types of primers and probe structures, you can see here the expected banding is the dark banding, of course, at the top. But all of this underneath those dark bands are faint bands that you can see here. Those are called primer dimers. Primer dimers is one of the oldest problems with the PCR technology in general. If you have low efficiency primers, you can get primer dimers. And the primer dimers, as you can see in the bottom picture, with the low efficiency primer, the dark banding here isn't even the expected amplicon, it's the primer dimer. So in some cases with a bad primer design or low efficiency primers, the primer dimer can actually have a stronger signal than the actual amplicon you're trying to find. So that would lead to a false positive result. Or in the case where it's you're not even looking at a band, maybe you have smears and stuff, or you've got very faint banding. It's all down to the human eye. Is the band there? Is the band not there? And, is it real or is it false? Is it a false negative? Is it a false positive? So the older traditional methods, it's possible to use them, but they come with a lot of problems, and they're also limited on what they can do. So in the case of this traditional PCR, we could multiplex like influenza and COVID together in the same test here, but then you got to look at the size and differentiate between the sizes. Is it influenza or is it COVID? And then with the more that you multiplex together, you even have a higher chance of producing these primer dimers. The flip side of this is you move towards more modern technology, real-time PCR being one of these. And you can see here this example on the right-hand side of the page is where they multiplexed RDRP gene E, N, and S in a big multiplex reaction. And you can see the dark blue amplification curve here is representation of the COVID test. And yes, that looks very nice, but you can see also there's a problem in this with these primers being all multiplexed together, where they're getting primer dimer formation in their no template control, which is evident in this purple amplification that's coming up towards cycle 40. So, once again, we're still dealing with primer design issues here. However, you do have a better test in that you can actively trace the amplification or a positive of COVID in real time. Now, let me switch to one of the technologies that Co-Diagnostics actually has. I'm going to switch slides here. Oops, not that one. This is an example of the Co-Primer, which is even newer, like 5th generation real-time PCR probe technology. And with this technology, its structure specifically puts it in a situation where it inhibits the formation of primer dimers. So we're overcoming some of those traditional problems that has been faced. And so we no longer have the false positive amplification, even when we multiplex up to 14 targets in a single reaction. So in here, you can see the structure of the co-primer is not like a single, just like TACMAN structure, which is 5' to 3' with just a sequence, a fragment. But here you've got a capture sequence, which is a longer genomic sequence, about 20, 30 base pairs, which is connected with a linker to a very short primer sequence. And so the structure itself helps the prevention of the primer dimers, because this capture is going to bind down to its target DNA first, and then the primer will bind. And as the polymerase extends through, it's going to extend through the primer, and then it'll extend through this capture, and it will stop where there's a carbon here on the end of this 3' of the capture, and prevent that from extending on and creating any type of primer dimer issues. So this is just an example of how more modern technology can play a very big role in overcoming the problems that have been faced with, primer dimers from regular primer design, and overcoming some of the development issues, because a lot of the newer technology is integrated in its design with the bioinformatics. So like Co-Diagnostics has its own software, and a lot of that software is built on bioinformatics platforms that can help scan the genomes. We can take the 22,000 COVID sequences that are in the database and scan those and keep monitoring them over time, so that we can see where we can build the test in the most conserved area of the genome, and be able to overcome the pitfalls of design.
So if this remains a bastion for more modern and more sensitive designs, can you speak a little bit to the audience about the recent developments in being able to just use saliva, and that way obliterate any kind of technician handling of patient samples when they are brought in? And would that be something that you see as gather momentum for large scale testing?
Yes, so the saliva testing can definitely gain some momentum, because it has the capability to mass test. So it's a non-invasive method, and definitely something that the patients would be more inclined to do. Like, I go to the doctor and I'm a very hard stick if they're trying to get blood from me. So I would much rather give them a saliva test
as opposed to a
collection
swab
sample, rather than having some swab stuck up my nose, or being stuck multiple times with the needle. So I see that as a plus side for the patients. And, it's also, it also helps culturally. I mean, there's some cultures and some religions that don't allow testing of blood samples, for example. So saliva would definitely overcome that. I think they would be more willing to provide samples and be able to protect themselves with any type of screening information they could get back.
The question is that does saliva give you enough DNA, I mean, in this case, RNA material to do a very effective test? And recent developments show that, because I've seen in the news reports about the school districts that have chosen that option to do large scale screening.
Yeah, so, I mean, typically, the COVID, it really depends on the pathogen. And right now we're talking about COVID. So, I mean, COVID is found in the respiratory system. And of course, your mouth is a big part of that. So the saliva would be there to, house and collect that viral material. So yes, I believe that the saliva would be able to produce enough RNA for detection, especially using some of the latest technology for the detection method.
Mm-hmm. Given that, how do you rate the recent spate of tests that have come in through antigen or antibodies? Decision has been constantly wavering between the two. for screening. So the mass assumption is that you take antigens or antibodies for a mass scale screening and then go for a confirmatory test with a PCR based test. So there are several aspects that influence that decision, maybe because, you know, technology, laboratory, the competence of the technicians, everything comes into play when you're talking about a PCR as opposed to what you do with antigens and antigen antibody. But where does that compromise us? Because the antibody tests had to be, you know, they've been virtually, US FDA has also kind of put a ban on it, or not a full ban, but they've retracted a few of their emergency use approvals.
Yes, so I think, you know, what people like about antibody tests is that they typically come in a cartridge and you can use them in the doctor's clinic. It doesn't take a lot of resources and manpower to have a cartridge, to put a sample on there and wait 5-10 minutes to get a result. Now, the problem with the antibody test for COVID is that I haven't really seen any convincing evidence that suggests that COVID antibodies are long lasting or that they provide any immunity. The publications that I've read, they show antibodies to COVID last only between 2 and 8 weeks. So in that situation, I believe that a real-time PCR test would definitely be the best option for detecting the acute infection. Acute meaning it's early detection. So like today I just started getting a fever, so I'm going to get tested by real-time PCR, and it would come back telling me a true answer if the virus is there or not, because during the first day of any type of symptoms, or even before symptoms sometimes, because there are people who have COVID who have no symptoms. I was one of those people back in May. I got tested. I showed positive. I had never had symptoms. I still haven't had symptoms. So during that period of time is when the viral RNA would be the highest. Let me quickly show you a slide that can show you the difference between antibody testing versus PCR testing. So this is a picture that I took from LGC. They actually bought out the native antigen company. And LGC, they're linked to Liu et al. And Li et al. Publications if you want to look up the full publications for these. But this is an example of where viral RNA comes up first. So you would be able to detect the viral RNA and then the antigen somewhat a little bit later than the viral RNA. And then this lasts, I think, between 12-15 days. And at that point in time, your body starts producing antibodies. So as your body starts to produce antibodies, of course, the viral RNA decreases because your body's trying to clear the viral RNA out and is producing immunity at that point in time. So the IgM antibody, of course, would be the first antibody that's produced. And then IgG would be a very longer lasting antibody that would be produced. So an antibody test in general tells you if you've ever in your past had that disease. Whereas the viral RNA test for like real-time PCR would tell you, how much you have right now. And not only could it be used for screening, but it could also be used quantitatively so that it helps doctors tell you in the end, how much medicine do you need to take based on your viral load. And that's why viral load testing for like HIV and hepatitis is so important, is because the physicians can use that information from the viral load to determine how much antiviral therapy, or even bacterial wise, how much antibiotic may be needed to treat that specific disease.
So now I think I will bring in Mr. Sarabhai for a few more questions related to the larger implications. Mohal, where do you see the growth of the market spreading out? Is it going to be in rapid testing, which I mean is in the antigen antibody, or you see that the high-end PCR is here to stay?
See, first of all, I appreciate the comments of Dr. Garcia and what you have said. And plus, we have a company, just for clarity, that is into RT-PCR. So that from a business side, that's where it is. But when I look at it as a citizen of India, it's not possible for us to test the way RT-PCR because of the limitations of the RT-PCR test. We understand that this is the gold standard. If you had any hospital today is only relying on a PCR test. If you want to get tested, if you have symptoms, they eventually rely on a PCR test. But we also need the country, we also need our world to get back to normal. We need to get back to students being able to go to school, employees being able to go to work, travel needs to happen. I mean, a lot of things need to happen. And for a country which such large population, such large clusters, a very accurate rapid test is definitely need of the hour. And that's what the government is doing. The government is testing in clusters there. You don't even have to pay for it. Yesterday, they came to my factory and tested 150 people on their own. We had to just, there were these rapid the antigen tests, like Dr. Garcia said, the result is within minutes. I know from a science perspective that they are not as accurate in the sense that if it's positive, it's most likely going to be positive. But if it's a negative, it doesn't mean that you're not positive. So if you're negative and you're showing symptoms, you do need a PCR test, which is the gold standard. But that still is giving us according to me, a large percentage of let's say safety, because you are being tested by by the antigen test. So I feel that from a market perspective, there is enough room for both these tests. Look at the numbers. We have 25 million people who are tested positive in the world. We have 900,000 people approx who have died so far. This disease is not going away until a very proper vaccination program is not only found, but implemented across the country, across the channel. Until then, there will need to be tests. I mean, there is no way, I don't see anything around it. So whether it's a 5-minute test or an antigen test, or what's eventually what everyone is saying because of the science behind it, that you do need a PCR test. So it depends on how we look at it, but there's a huge market for all these tests, and all these tests are going to be used. Another thing, what has happened, at least in India and some of the places, is that because of the test, the PCR test was also very expensive when we started. When the COVID thing started, a PCR test was very expensive. You would see now that in India, the PCR testing has come down drastically. The price, both for the test and as well as what they charge the patient, has come down drastically. So eventually, from an economics perspective, all these tests would be relatively in the same sort of area. But the speed that an antibody test would be required for mass screening, and I think for confirmatory tests, a PCR test would be needed. So I think both the tests rather have a huge potential, and we need to get a very accurate antigen antibody test as well.
Do you think that even despite the vaccine, I mean, this question probably is directed to both of you, is that once you have a vaccine, it depends on what kind of immunity the vaccine, even a good vaccine, is going to confer on a patient, or so you still see vaccine going in tandem with regular testing?
I get this question, Dr. Mayuranki, I get this question a lot. In fact, because of the business we are in,
Right.
But look at where we are. Even if a vaccine is available, let's say in India, forget the world, let's say a vaccine is available in India first quarter of 2021. By the time the vials are produced, the vaccine is implemented across India, we are talking about a long period of time, especially now that people are saying, or science is saying, that immunity, how long does it last? Dr. Garcia is saying 2 to 8 weeks. I've heard figures that go to 3 to 6 months. Nobody today is saying 1 year or forever, which is what one would like. So even how is this vaccine going to even work? Do they need another dose of vaccine after 6 months? If that is the case, it's even longer. So the idea is that the testing will have to remain, because once you open up, as I said, schools, universities, travel, people would need to be tested. So vaccine or not, until at least for the next 24 months, I see we have to test as much as possible, because even one positive person we have found everywhere in the world can infect so many people, especially when people are asymptomatic. It's even worse. So we take all the precautions, but at the end of the day, it's testing, testing, testing for the next 2 years. There's no way around it, according to me.
Yeah, I agree. I think, vaccination isn't foolproof. I mean, we know that from the influenza vaccinations. You can still get the flu. So I think the same will be with COVID. I mean, you got to think COVID is coming from a group of coronavirus that caused the common cold, and everybody gets the common cold. So even if there's a vaccine for it, I think we're still going to have testing. And, I also agree that you have to have companion testing. The RT-PCR has to be combined with the antibody tests. That gives you the full picture of, are they currently infected, or do they have a longer term immunity? If I always am a proponent of companion testing. So I believe that is needed. And I also think that the industry needs to work towards getting pharmacogenomic testing in place, so that whenever there is a medicine like Remdesivir or some other medicine that actually can help, we will know which patients should receive that treatment or not, based on what their genetics is going to say work. Will better process how they metabolize that particular medicine and how it will affect them, because we definitely don't want to end up vaccinating or either treating patients with something that's going to cause them long-term side effects,
I agree.
in the future.
And I would just add that as doctors understand, or as the medical field, as pharmaceutical companies understand more about this disease, the number of deaths is also decreasing, because we know now which drug to give for which symptom, and we've got a full understanding of this. There are very even low, I would say, low priced drugs that are working, and that we're hearing this from all over the world. And that's an equally important or very important step, that if even the death rate goes down to very negligible norms, you would have won this battle, or you would have stepped ahead of this battle with COVID, with drugs that are already available in the world today. So a lot of research has to be done there also. That was just my addition to this whole thing. Besides there's testing, there's vaccine, and there's also medicines that playing a big role.
You obviously think, Mr. Sarabhai, about the larger implications, and that is why you may have must have informed your decision to move into diagnostics. How do you see the role of COSARA as we grow along, in terms of, what you know of the in terms of COVID specifically in Asia and Africa and in South America, given what's happening over there in Brazil, in Central America also?
See, I may be off by a few months, but when we started COSARA, COVID was not there. And even if you look at today's numbers, and quote me if I'm wrong, someone correct me if I'm wrong, rather, I think TB, which is what we started this company for, tuberculosis in India is killing more people than COVID, even if you consider the quarter numbers. You know, it's slightly higher. It's reaching up, but it's slightly higher. TB and you know, PCR testing, what we just discussed, it's something that we need to completely eradicate out of this country. And that was one of the first reasons that historically my grandfather had started the medicines for that, the actual treatment for that. We were the first to bring streptomycin into the country. Then the diagnosis was very poor. So a lot of people were just not being diagnosed correctly because there was drug-resistant TB. So the genesis of this company started because we needed a cheap, not a cheap, I would say economically priced, everybody should afford it, molecular test with PCR for TB, and that's what we did. And now COVID has come, so we have that. And we've got a lot of other infectious diseases, like right now, chikungunya and dengue have started in Gujarat at least. So I got a call from someone saying in Gujarati, saying, "Oh, koi vandho nahi, COVID nathi, dengue che," you know, as if it's a good thing to get dengue. But dengue is also equally not so good to have. But this is the sort of philosophy of what it is, and we have tests for that distinguish between dengue and chikungunya, the multiplex test we are saying. So that was first part of the questions. I think we have a good diagnostic company that looks at infectious diseases happening in India. And we've done that, and we have all those tests available. From a global perspective, what has happened is that, you know, most of these countries, at least in Asia and Africa, have been dominated by Chinese and Korean companies for diagnostic tests. I mean, that's 90% of the market.
Very true.
India has not been there particularly. But because of COVID and sort of the report of COVID worldwide is that India has done I would say fairly good job. There are obviously going to be people who don't agree with it. We've done a fairly good job. And a large part of that, besides the lockdown and the government schemes, have been that India has been able to give its own testing, its own diagnosis, and soon its own vaccine. So suddenly in this region, India is now saying, oh, even these guys have good diagnostic kits. We are being known. We have our companies, not only us, there are about 5 or 6 other companies are known as diagnostics. So there is a lot of interest of our tests going into these Asian and African markets because they don't have their own local test. And today, nobody wants to rely on China. I think China has, after all this, rightly or wrongly, people would much rather buy an Indian product than a Chinese product because we're also well priced. So I feel that a great potential for all Indian companies to go into these markets with all the molecules. Of course, right now the export is not allowed by the Indian government fully, rightly so, because it first wants to take care of the Indian citizens. But this is going to change soon because once we get more and more production out, and we will be exporting to all these countries as well.
Talking about what you and Rebecca were discussing about what actually some patients find themselves more vulnerable to a COVID attack than others. Now, in a recent article in Nature, Rebecca, they were showing that the ORF1AB gene variants and the S protein, the spike protein variants, were linked to high mortality. Of course, this study, like everything else in COVID, doesn't have a very long precedence, but there was some data that Nature published in this study. And the other connection they were trying to draw was from the BCG vaccinated populations and their susceptibility to COVID. Can you share some information with the audience on that?
Well, I think the correlations that they made in that publication open the window for more studies. There's definitely a need for more studies to see if vaccination plays a role in, you know, the ability of one to get COVID or be at a higher risk, you know, or even the extent of the symptoms they exhibit. But overall, the data should be integrated into improving current test designs and development of the new tests. I think more information is definitely needed in addition to the genes being linked to a high mortality, as there are variables which could be involved, like a high viral load associated with the mortality and the role that underlying disease plays in gene expression or genomic variation. For example, the high viral loads of COVID have also been shown in peer-reviewed publication to be associated with age, comorbidities, treatments, and smoking. And the viral load has been independently associated with mortality rates.
So you have a slide, I think, that talks to that, right? I mean, some of the scar tissues.
Yes, I do have a slide. Just a second, I'll pull it up. Let me share my screen. So this is a picture of the scar tissue that can build up. It's a picture, a CT image of the lungs for COVID patients. And this one I took from Pan et al. Publication from radiology, but there's several that show exactly the same types of pictures. And even from early on, actually, when I was going through the literature that was published between January and April of this year, and most of that came out of China, where they had started, their initial testing plan was CT imaging of patients along with the real-time PCR broad testing. And what they found was that most of the chest findings, whether they were asymptomatic patients who tested positive by real-time PCR or whether they were actually patients who had been put on a respirator, they found that they had some of the same types of imaging of their lungs. And you can see in this section A here, you see the little bright white looking things that are in the dark color on the right side of that picture. That's the lung tissue. And so what those little white things are they're called ground glass opacities, or GGOs. Those are very common with all COVID patients. And then in the second one, you can see this one is got a little bit more involvement in the lung, and C, of course, has a lot of involvement. So in the picture B there is called a crazy paving pattern. So you've got a little bit more of those more of those little white squiggly lines in the lung tissue. And then, of course, you've got starting a thickening of the septal around the lungs there. So you can see that. And then, of course, in picture C is complete consolidation. So the patient in C, that patient would probably be on a ventilator because their lungs are not going to be able to push the air out because they're so damaged. But and this would, the C, as the lungs get more and more damaged, it's hard to recover and come off of the ventilator. So, that leads to a high mortality as the lungs get damaged more and more throughout the progression of the disease. And actually, a lot of times it's not just an increase in virus that causes this, but the body's actual immune response. A lot of the problems with the COVID cases is that their bodies go into a hyperactive mode of response. And so that's what's creating all the problems, because once your immune system gets so stimulated and starts creating all these immune responses, that's where you have the breakdown of all the organs is possible. So
before we sign off and let the floor be open for other questions, I have a last question for Mohal. So does this look like there would be a very good market for a saliva based testing here in India, because it simplifies the collection?
Absolutely. I feel that, another whole issue of the current method is that whether you ask for a PCR test at home or you go to the technician himself is coming with a PPE suit. He's doing so many tests, he or she, probably not even wearing it properly. The the suspected COVID patient has to remove the mask,
Right.
stick a swab in the throat, in the nose. So it is so risk to the healthcare worker. And they do large amount. I checked with one healthcare worker who had come into my area, and I mean they're doing this. They're collecting 50, 60 samples in a day. These are people who go around, and because the labs are sending these people out. Each time you're supposed to wear a new PPE suit, but I mean, how many people are doing it? How many people are trained to do it in such a way in this heat, going in a scooter or someplace like that? So I feel that it is just completely not a workable situation for a large scale testing if India really needs to get out. So a saliva test, which is, I do it myself. I'm assuming I read a report in the Yale University has developed where you can just spit and you can collect the thing. You don't even have to have any lab technician whatsoever. You can send that sample just like we are doing a urine sample or something like that, and then it goes into the regular testing and you get a result. It just saves so much of the hassle. It's so non-invasive. It's so much of a hassle that I feel it will be a great game changer in India, and we are working on that as well with our partners, CodeX, to show the sensitivity and the specificity with the saliva sample. But I think it would be a game changer, especially in India, and plus for a patient, it's very uncomfortable to have these noses. It's not here. They take it right from here. That. So extremely uncomfortable even for the patient and overall. So saliva, I think it would be a great thing if we can get it validated as soon as possible. And we are trying that as our company.
So
we are trying that.
Yes, so that because Co-Diagnostics in the US has already supplied the reagents for it, and the central reference labs have already gone ahead and got an FDA approval on it.
Correct.
So I'm sure you should be looking at that.
We are absolutely looking at it. We absolutely see the great potential for that. It's more tangible potential than some of the other things that you hear in the paper that, that they look in something and you get it in 1 minute and 2 minutes and everything. This is something that's a proven technology.
It's more realistic.
It's so much more realistic, and it's a next step for us, And I don't mean for us, I mean for the country, for the world.
From the laboratory side of this, the saliva sample actually makes it where we don't have to use extraction. I know the problem has been obtaining supplies for extraction. Even if we can make 1 million PCR tests, if we can't get the extraction, that creates a blockade for us. But the Yale study is very interesting, and we've been testing at Co-Diagnostics in Salt Lake City, where you're basically taking the saliva, adding proteinase K and boiling it and putting it direct to PCR. So it completely alleviates the need for the extraction. And that would definitely be a move forward in being able to mass test.
Correct.
So I think from my side, the questions have kind of covered the whole comprehensive overview. And I would request Dr. Bakshi now to open it up for queries or any further discussions targeted to any of the participants. Thank you very much.
Thank you, Dr. Mayanki.
Thank you.
Thank you, ma'am. And yes, there are a few questions which have been posted in the chat. So I will just read out one by one. The first question is from Dr. Parth Pandya. What is the rate of mutation occurring in SARS-CoV-2 compared to MERS-CoV and other corona family virus?
That's a Dr. Garcia question.
Yes, and unfortunately, I don't have that information right here with me. I just know that the COVID-19 virus, even though it's mutating, it's not mutating a lot. It's actually more stable than some viruses. But in general, I think I haven't seen much information about the difference of mutation rates between the viruses. So I can't really answer that question.
Okay. We can go to the next question, which is from Professor A.V. Ramchandran. Sir wants to know regarding vaccine development, how far, or for that matter, how long would a protein-based vaccine be effective, especially considering the COVID genome being very pliable? Shouldn't we go for more RNA-based vaccines?
I somewhat agree with that statement. I really think, the vaccine, if we could do some type of RNA-based vaccine, I think it would be better because we're dealing with the RNA virus. I think that also extends to treatment. I know whenever everybody was saying, oh, the malaria medicine will work for coronavirus, I'm like, what? Malaria is a parasite. We're talking about a virus here. That doesn't even make sense to me. So, I think, whenever you're looking at a specific species or you're looking at, a virus versus a bacteria and such, if you stay within the same type, I think it has a more effective treatment and a more effective possibility for vaccine. But in any rate, I'm not a person who's in the vaccine development world, so I don't have a lot of information to provide there. But I do know that, whenever we have a vaccine ready, it takes at least a year or two to go through all the clinical trials that's required to make sure that it's going to be effective. And I know from what I've been reading, there's a lot of hesitation, and worry now that we're going to now release a vaccine just because we're in a rush to do so, instead of making sure that we've done all of the thorough research that needs to be done. And I know here in the States, there's a lot of different programs where you can sign up to be in the vaccination trials. Even in Atlanta at Emory University, they have a big vaccination trial there. But they're giving these people the vaccines and they're like, just, go about your business and wear your mask. Are they really going to be exposed to the virus anyways? There's a lot of questions there. So we'll see.
But maybe Dr. Ramchandran's question was also aimed at, it is probably easier to go the protein way than to go the RNA way to make the vaccine.
Yeah, it probably would be easier. I think one's as hard as the other. But the protein typically in test design, from my experience, protein is somewhat easier than doing genomics.
Yes.
But isn't Moderna's vaccine RNA based, Dr. Garcia?
I'm sorry.
is.
The Moderna's vaccine in the US that got the US funding, I think, is RNA based.
It is RNA based, but that's why it's not it's not promising fantastic timelines either.
It's phase 2 right now. It's phase 2, yeah.
Right.
Yeah. So people, to answer that, people are working on the RNA vaccine as well.
Correct, and they may be the one to last. Yes.
So I think Pfizer also is working on it, on the RNA vaccine. So it is there. It is just that who comes first, and there's also, it's political also as to what.
Very. Yes.
Extremely as to who's funded what and
especially
what it is.
In an election year.
Yeah. So.
Yes. That's true.
Yeah.
Okay, we can now move to the next question. This is from a fourth-year student of ours, Arnav, from the Biomedical Science MSc program. And he has a very specific question. He says, in COVID-19, cathepsin L mediates the cleavage of the S1 subunit of the coronavirus surface spike glycoprotein. Can the levels of cathepsin L be somehow be used in diagnosis, or can we anyway use the same for the treatment?
Treatment seems to be more likely.
Okay.
Because this is a hard thing to actually predict in terms of diagnosis. It works, but how do you increase it to the point that it can really be used as a diagnostic is difficult. It will be longer but easier to use it as in the process of treatment.
Okay. Another question from the same batch of students, Bhavya Gupta. She wants to know, one of the lessons we learned from COVID is that how important diagnostics are, not only to detect the patients and treat them, but also prevent its spread. what are the diagnostic strategies we should target in the future to prevent such outbreak?
Wow, very loaded question. Rebecca, you want to take a stab at this?
So, well, you know, the real-time PCR would be what is recommended for prevention of spread, because it's the first one that would be playing a role in the detection of the virus. But utilizing diagnostics for prevention of disease, you know, the way to do that would be to look at the risk factors. And there's a lot of controversy when you're looking at risk factors. I know Map My Genome in India has done a lot of testing for risk. You know, what puts you at risk for diabetes? Well, what puts you at risk for getting COVID? I mean, so those would be a lot of different things to look at if you're building a prognostic type of test. But it's definitely, I think, something that should be looked at. I mean, and the other thing is you get into an ethical debate. You know, if we do create these types of tests that look at risk factors, then to what degree can those be used at, you know, allowing you to get health insurance, or allowing you to work, you know, do certain jobs, or, you know, work with certain chemicals, or even, you know, certain degree programs that might require your exposure to things if you're more prone to, if you know, if that exposure is going to lead to a greater risk factor for you.
Okay. The next question is from Dr. Lipi Butch, and she wants to know what is the percentage of getting a false positive or false negative result with RT-PCR?
Okay, well, if you look, I'm going to go by the FDA guidelines in the US because they're somewhat the same globally. If you look at like the European standards and such, the FDA actually recommends that all tests be validated before they're used. And of course, that includes all the validation data that would be put into a submission. So even with like the CDSCO, ICMR, we have to submit a technical file that has all of that validation data in it. And the FDA here in the States provides recommendations. It's in section 5 of the policy for COVID. You can find it, if you Google it, you can find it from the FDA website. But it says that for molecular tests, mainly referring to RT-PCR, but if the molecular test is like LAMP testing, or if it's some type of molecular antigen testing, it would fall in the same category. They say that the limit of detection is required and may be done using spiking RNA or inactivated virus into artificial or real clinical matrix samples. And then they go on and they define the limit of detection, which is the LOD, as the lowest concentration at which 19 out of 20 replicates are positive. So that's 95% positive. So if the requirements from the regulatory groups are that the test has to be 95% positive, then you got to look at that mathematically, statistically. If it's 95% positive, then there's a 5% chance there could be a false result out of that. So whenever they're saying 19 out of 20 replicates, and you've actually tested those in the lab, well, if you've got 19 out of 20, that means you've got one that didn't give you the correct answer, or what you were expecting, let's say that. And then for that one, you need to go and see, is it a real answer or not? So like for real-time PCR, if you had one sample that wasn't what you were expecting, then you typically have to sequence that, because the sequence is going to be the end all be all result of, is it really a COVID negative or a COVID positive, depending on which way it went.
Okay.
They just to add that in India, the ICMR guidelines actually show 100%. If your test needs to be approved, it's 100% specificity and sensitivity. So out of all the samples that they test, all of them have to come with the same result. That's what the guideline is for getting approval for any test that's sold in India. It's a very stringent, whether it's even possible is a debate, but that is the actual standard for India. So any test you're getting here, a PCR test, has to match that sort of standard. So it's more stringent from a law perspective than what the US is doing.
Well,
Okay.
just let me clarify. So the limit of detection and then the agreement between one test and another are actually two different things. So when I'm saying the limit of detection, I'm talking about a known concentration of virus that's been diluted into a serial dilution, and that gives the limit of detection. But if you're looking at clinical evaluation, where you're comparing one test to another, there has to be a 95% agreement at a 1x or 2x limit of detection and a 100% agreement at all other concentrations. So it's only the very low end of the test that has the 95%. But once you're above that 95%, just like, for example, the co-diagnostics test has a 4.29 copies per microliter limit of detection. So anything above 4.29, like let's say 5 copies per microliter, needs to consistently give 100% sensitivity, specificity for an agreement with other tests.
Okay, continuing to that, there's a question from Dr. Sandeep Patil, and he wants to know what are the, how does a government agency approve a diagnostic kit? And how does one ensure that the test results are correctly inferred? I think this is also in context to India or US or comparing the countries.
Correct.
No, that's what I was mentioning, I think,
Yeah.
when I said that if you're a manufacturer or an importer or whichever way, if you want to sell a product in India, again, I'm saying in India, you have to send it to one of the ICMR approved centers. So there are a few right now. There's one in Pune, there is one in Delhi, there's one in Kolkata. They've approved these centers where you send the test. They have their own standard that they say that this is my gold standard. And the test you send, they will run it with all the samples that they have. And it has to show 100% matching with the positive and the negative. And once you do that, and obviously in addition to that, all your manufacturing documents and what you use and how many genes and everything, once eventually this happens, then they give you an approval for sale into India. It's a very stringent process, rightfully so.
Yeah, and I think it's like that for any country. I do know that, any country is going to rely heavily on that technical file that you submit, and that technical file is going to be all the data that's been collected for limit of detection, sensitivity, specificity, cross-reactivity, inhibition studies. So you're going to submit all that data, and, once you submit the data to the government agency, they're going to look through everything and see if they agree with your analysis. And they have statisticians and mathematicians that can go through it, and they look at what you've collected. They may even ask for the raw data files so they can analyze it. They may ask for additional data, like if you've run 20 replicates and they're not convinced, they're going to ask you for another 20, 40, 50 replicates. And so they're going to compare that, and they're going to compare your test not only with the data you collected, but with other like things in the market. They typically refer to those as substantial equivalence. So if you've built a test that's RDRP test, then they're probably going to compare it to other RDRP tests in the market. They're not going to compare things that aren't similar in their methodology and technology.
Okay, I think we can take a few last questions. There are actually many questions, but I'm sure we don't have time to cover all the questions.
Yeah, I have to
So
be somewhere at 8.
yeah, so there's one question from a third year microbiology student, and she wants to know what is the safety of the plasma therapy for treating COVID-19 patients? How safe is it?
Safety. Okay. I thought effectivity is a question mark, but.
So the safety of plasma would be the same as any type of transfusion. You run the risk of having like a transfusion reaction where the plasma has to be typed, not only on an ABO typing level, but also you got kid kill Duffy antibodies that have to be typed. So you always run the risk of having some type of transfusion reactant, whether it's just a low grade fever or whether it's actually ends up being more severe than that. So there is that risk that's involved with plasma therapy. And then of course, if you continually have any type of plasma type therapy, you run the risk of acquiring some of that donor's antibodies. So over time, that can actually change the antibodies that you have in your own system. And that's what, of course, they're hoping will be effective for the COVID treatment, because you're acquiring the antibodies that that patient had to help them get better from the disease. But over time, it could also lead to other potential risks for other diseases or other complications. So, I mean, all of that has to be considered with the treatment.
Okay. I think there are a few more questions, but as I think we are already running over time with the session, so I would like to request Dr. Elizabeth Robin to propose the vote of thanks.
Thank you, Dr. Darshi. Okay, so now we come to the end of an enthralling and captivating session. On behalf of School of Science, deem it a great honor to propose the vote of thanks to all who have helped us in making this conversation series such a resounding success. First of all, I would like to thank our esteemed speaker, Dr. Rebecca, to accept our invitation to participate in this conversation. Thank you, madam, for giving an insight of whether PCR is a final answer as a part of COVID testing. I would also like to extend my thankfulness to Mr. Mohal Sarabhai for sparing his valuable time to talk of the technology behind the COVID testing. Sir, we would like to have such intriguing sessions with you in the future also. Thank you so much, sir. Thank you, Dr. Mayuranki, for moderating this session and asking so many intriguing questions to our esteemed speakers. I would also like to extend my sincere gratitude to Nilay sir and Ramchandra sir for their moral support and guidance. I also thank Dinesh sir for the technical support. I also thank all the attendees who have participated overwhelmingly and making the event a success. So I would like to meet you also in the coming sessions too. Thank you so much.
All the questions that have not been answered, we request everyone to post them to us, specifically the students, and in future times we'll get them passed on to our speakers and get the answers. Thank you. Thank you for posting.
Thank you. Good night.
Thank you very much.
Thank you, everyone.
Good night.
Thank you.
Thank you.
Thank you very much. Thank you. Good night.
Bye-bye.